Hello, and thank you for coming to our symposium. My name is Amanda Lyall, and I'm an assistant professor in the Department of Psychiatry at Harvard Medical School. I am sorry that I cannot be there to share this work with you in person, but I hope that we can find some time to discuss this work in the question and answer session after. The title of my talk is Attaining Novel Insights into the Biological Correlates of Schizophrenia Using Free Water Imaging. I have no disclosures. As we have learned through the preceding two talks, there are a large number of studies that have used diffusion tensor imaging in populations of individuals with schizophrenia. This large body of work has strongly implicated white matter as a central pathology in the etiology of the illness. Here, I highlight two principal studies which showcase this. One, which is the Enigma study by Kelly and colleagues, and the other is a study by our group, which was discussed in depth earlier, both of which show that the diffusion tensor metric fractional anisotropy, or FA, is reduced in individuals with schizophrenia. However, despite the power of using diffusion MRI approaches to study white matter microstructure, there are some limitations to the traditional tensor model. The principal metric FA, while highly sensitive, is not very specific. This can be best visualized in the figure on the right, which shows that the large number of tensor shapes that can all produce the same FA value. The FA signal can be influenced by several things, both biological and pathological. These can include biological or inter-individual differences in axonal density, underlying tract geometry, and cell packing, and then can also be affected by pathological changes like edema, glial scarring, and demyelination. Thus, there's a great need to develop new models for diffusion data that can provide more information about the underlying biological contributions to the diffusion signal. One example of a new model is free water imaging, which was developed by our colleague, Dr. Ofer Pasternak, in 2009. In this model, the diffusion signal is deconstructed into two compartments. The first measures the amount of unrestricted water diffusion in each voxel, which is modeled as an isotropic FEAR, what we term extracellular free water. The other compartment measures the amount of restricted water diffusion that is in close proximity to tissues, which is then modeled as a tensor and which we call fractional anisotropy of the tissue. This produces two whole brain maps, as you can see on the right, which are used for future statistical analysis. We have previously applied this approach to a few populations of individuals with schizophrenia in the past. To summarize a few of our published studies, we have shown that in early stage patients, they exhibit significant global increases in free water, as shown here in the left in blue. More importantly, we show that these increases in free water highly overlap with the significant reductions that are found in FA when we use the diffusion tensor imaging model separately. This may suggest that the reductions in FA that we see in early stages may not necessarily be indicative of aberrant white matter microstructure, as has been previously argued. Interestingly, this increase in free water is not seen in chronic subjects, which suggests that this extracellular is relatively diminished in chronic subjects, which suggests that this extracellular pathology may diminish over time. However, we still don't have a complete picture regarding the trajectory of free water changes over the illness course. This is because the studies that have utilized free water imaging in schizophrenia have only ever studied relatively smaller sample sizes with very discrete illness windows, such as only focusing on early stage or chronic subjects. Up till now, there has not been an avenue to apply free water in a large, age-diverse sample to directly examine the relationship between free water and duration of illness or age. We were able to overcome these limitations in this study by leveraging the retrospective harmonization approach discussed in the previous talk. Our study aims are, first, to determine the trajectory of age-related free water changes in individuals with schizophrenia compared to matched healthy controls. And then second, to directly examine the relationship between free water and duration of illness. Based on our previous work, we hypothesized that independent of the effects of age, individuals with schizophrenia who have a shorter duration of illness will show the greatest free water differences relative to individuals with more prolonged illness. Our sample includes the diffusion MRI scans of 889 participants from 12 international sites. All diffusion scans were acquired using 3T scanners. Out of the 889 participants, 497 of them were diagnosed with schizophrenia across a wide range of illness durations, as shown here. 392 were included as healthy controls. For this study, we tried to conclude the controls whose age and sex distributions matched those of the individual's schizophrenia as much as possible. While I won't go into too much detail about the image processing, because this was already discussed at length in the previous talk, all diffusion data was minimally pre-processed across sites with consistent pre-processing steps. Then we applied our harmonization approach, followed by the free water imaging, which created the free water map for each individual. For the final step, we calculated a whole brain average free water value for each individual only in the white matter, and then ran trajectory analyses for a 40-year period separately for controls and patients using a non-parametric sliding window analysis. What we see is that in controls, which can be shown in orange, we see a relatively stable pattern of free water values across the 40-year period. We also see that across the entire age range, controls exhibit lower free water values than individuals with schizophrenia, which can be seen in blue. Within the patient cohort, we see that young ages are characterized by higher free water values, which decrease slowly until the age of 35. After 35 years of age, free water values begin to increase again, but this increase is at a relatively slower rate. When we look at the effect sizes of the differences between the patients and controls, we can see that the effect sizes are greatest at the younger ages. We also see that there are significant differences between patients and controls between the beginning of the time period and until the age of 35. Between 35 and 44, there are no significant differences between patients and controls, but these differences begin to emerge again after 44 years of age, by which free water values start to increase and the differences between patients and controls begin to increase again. Finally, to investigate the relationship between free water values and duration of illness, we transformed free water values into z-scores using the unbiased estimates of the mean and standard deviation for the healthy controls. These z-scores were then used to investigate the relationship of free water and duration of illness. We see that there is a gradual decaying trajectory such that those with the shortest duration of illness had the greatest deviation from the healthy mean, which this analysis was also corrected for age, sex, and current medication dosage. So what we found pretty simply is that free water is elevated across the entire illness course when you compare them to healthy matched controls, but that these greatest differences are found in younger individuals with schizophrenia. Moreover, we confirmed our hypothesis that higher free water seems to be related to a shorter duration of illness. However, what biological information do these free water changes provide us? One possible option for free water differences could be tissue atrophy, as this would lead to an increase in unrestricted water signal. However, previous imaging studies, such as those using structural MRI, typically do not report widespread white matter tissue atrophy in early stages of the illness. Instead, white matter tissue atrophy is more commonly observed in older subjects, and therefore could explain why we see an increase in free water in older subjects or those individuals after the age of 44. But what's taking place in the younger subjects? Another alternative is that it could be indicative of immune activation. There is some converging evidence to support this. In this study of an Australian cohort published by DiBiase and colleagues, we see that there are significant positive relationships between free water values and key pro-inflammatory cytokines, specifically IL-6 and TNF-alpha, such that higher free water was found in individuals with the highest levels of these pro-inflammatory cytokines. Similarly, there is preclinical evidence for the involvement of the immune system as well. In this rodent study, which employed the maternal immune activation model, male rats prenatally exposed to the viral mimic poly-IC showed elevated free water relative to their saline-treated counterparts at postnatal day 90. As of yet, there has not been any direct evidence linking free water to immune activation, but this is an area of active investigation. So to summarize this talk, this study was able to expand on the current literature through the use of retrospective harmonization, allowing us to investigate free water trajectories in a large, age-diverse population of individuals with schizophrenia. We found that free water was elevated across the entire illness course, but as we expected, showed the greatest increases in younger patients with a shorter duration of illness. While this is still being investigated, we believe that there is a likelihood that these free water elevations are indicative of immune activation taking place at the early stage of the illness, whereas we believe tissue atrophy is likely explaining the free water differences we see in patients older than 44 years of age. This study has a few limitations, but one key limitation is that the data we harmonized was entirely cross-sectional. It will be necessary to replicate our study again with longitudinal data to understand how free water evolves in an individual and across different individual manifestations. Finally, this sample included only individuals with schizophrenia, but important future work should also see whether free water changes are present in other psychiatric disorders and determine whether or not it is specific to those with psychotic pathologies or not. I'd like to thank everyone who made this study possible, in particular the co-first author on this manuscript, Dr. Chetin Kariyamak, our Boston-based team, and all of our collaborators. We'd also like to thank our generous funding agencies. Thank you for listening, and I look forward to taking your questions shortly.